안녕하세요. 소개 정말 감사드립니다. 먼저 이렇게 바쁘신 와중에 제 웨비나에 이렇게 참석해 주신 여러분들께 진심으로 감사드립니다. 저는 싱가포르 국립대학교에서 박사학위를 취득한 임수빈이라고 합니다. 본 연구 발표는 영어로 진행될 예정이고요. 질문은 한국어나 영어로 편하게 해주시면 감사드리겠습니다. 그러면 연구 발표 시작하겠습니다. So this work was done at National University of Singapore as a part of my PhD research. So here I'll be discussing how we can make use of microfluidic devices as well as big data analytics to analyze circulating tumor cells at the single cell resolution. So cancer is the second leading cause of death worldwide, and metastasis accounts for the majority of cancer-related deaths. And so it simply means the spread of cancer cells to different parts of the body. So for example, when a tumor originates from a colon or large intestine, the cancer cells have the ability to travel to distant organs such as liver or lung. And this process can be done through either bloodstream or lymph system. So once the cancer cells successfully colonize at distant organ and proliferate and grow again, it will eventually form distant metastasis. So this is another schematic representation of the multiple steps occurred in metastasis. So the cancer cells can invade the surrounding matrix called extracellular matrix or ECM. The cancer cells can intravasate within the bloodstream. It means it can enter the bloodstream, travel within the bloodstream, and eventually extravasate and proliferate and colonize at the secondary site. So understanding these processes are highly complex and heterogeneous. So understanding the metastatic cascade at molecular level is critical to fight against cancer. So tumor heterogeneity can occur in different steps of the metastasis, and it is currently a major challenge in the field. So for example, a homogeneous tumor will be easily eliminated when a drug targeting a specific mutation is injected, given that all individual cancer cells carry the same set of mutation. However, a tumor is highly heterogeneous, so although a drug may remove a subset of cancer cells, there will still be a subset of cells that are resistant to the drug. So eventually, these subpopulations of the cell will repopulate the whole tumor again. And this heterogeneity can occur at multiple levels. So for example, when there is a heterogeneity within the same tumor, it is termed as intratumor heterogeneity. And when there is heterogeneity across different tumors for the same type of cancer, it's referred to as intertumor heterogeneity. And when there is a heterogeneity across different cancer types, it's called intercancer heterogeneity. And of course, different genes can have opposing impact on prognostication. So for example, the presence of the green mutation here may alter the bad prognosis of red mutations present within the tumor. So while we all know that there is a high level of heterogeneity across different tumor and within the same tumor, its impact on prognostication is still not clear. So our initial aim was first to derive the prognostic gene panel and study such tumor heterogeneity. So we first initially aimed to derive prognostic gene panels using so-called big data analytics. So here we generated a merged microarray dataset, or MMD, to integrate multiple independent datasets that are publicly available at NCBI. So each dataset carries the information on transcriptome profiles of cancer as well as matched normal tissues. So here, when we use the principal component plot or PC plot, we can see the variation across different datasets. So here we see a very clear separation across the different datasets. And this means that there is a clear technical variation, so-called batch effect, across the different datasets that need to be adjusted before using it for further analysis. So we performed a combat statistical method where it adjusted such batch effect. And when we use the PC plot again and colored the samples by disease status and dataset again, we see a separation between the tumor versus normal. So here the blue points represent tumor tissues and the red points represent the normal tissues. There's another way to validate this merged dataset. So we can again compare our dataset to another gene expression dataset, which is TCGA, which is provided by National Cancer Institute. And so by comparing all protein coding genes and compare their differential expression, we see that there is a very high concordance between the ranked list generated by both TCGA as well as MMD. So here, the microarray derived dataset is our MMD dataset, and here the TCGA dataset is derived from the RNA-seq platform. So when we see the correlation is highly correlated, so it means that the ranked list of all differential expression genes are highly correlated. We can again use the gene set enrichment analysis. So here what we do is this analysis assessed if a set of genes that are highly expressed in tumors would also be highly expressed in TCGA tumor samples. So here, when we computed the enrichment score, it was about 0.9, which is close to 1. So it means that this subset of genes was indeed highly upregulated in TCGA tumor samples. So after confirming the validity of our merged dataset, we can then use this merged dataset for further downstream analysis. So we here performed the differential expression analysis where we compared again the whole transcriptome, and we found that about 103 genes were differentially expressed in tumors versus normal tissues. Sorry, I forgot to mention that here we are looking at the non-small cell lung cancer. So it is the most common type of lung cancer in this case. And so when we look at the differentially expressed genes in NSCLC or lung tumor tissues, we found that in fact, almost 1/3 of differentially expressed genes actually extracellular or ECM-related genes. So these ECM-related genes contain collagen, ECM glycan, proteoglycan, ECM regulator, ECM proteins, and other secreted factors. And all these ECM-related genes are collectively termed as metrisome. So metrisome is on ECM-related components. And in tumor, besides tumor cells, there are other types of cells as well, such as recruited fibroblasts, bone marrow-derived cells, other immune cells, such as macrophages. And there's also extracellular matrix that provides either structural or biochemical or biophysical support to the tumor cells. And there are other factors such as growth factor, secreted factors, cytokines, and chemokines. So we first hypothesized that those 29 differentially expressed metrism genes may be used as a tool to predict survival outcomes of lung cancer patients, given their differential expression. So here we developed our own scoring metrics termed tumor metrism index, or TMI, which is computed based on the expression levels of 29 ECM genes or metrisome genes. So this is a heat map which visualizes the relative expression levels of genes. So here, these genes are 29 metrisome genes, and we computed the tumor metrism index for individual just using this 29 gene expression level information. And from the left to right, the patient has higher risk score or higher TMI. And when we just use this TMI and stratify the patients into two groups, low risk group and high risk group, we found that these two groups had significantly different survival outcomes. So we have done extensive validation using multiple independent data sets comprising more than 2,000 NSCLC patients. And we found that TMI was prognostic of overall survival as well as recurrence-free survival. And more importantly, it was predictive of response to adjuvant chemotherapy. So here again, by just looking at these figures, we see a very high level of inter-tumor heterogeneity. So it means that even for the same subtype of cancer, which was non-small cell lung cancer, there was a wide or very high level of inter-tumor heterogeneity. And although this is beyond the scope of my presentation today, using the same bioinformatics workflow, we have generated merged microarray datasets for 11 types of cancer. So these datasets are now currently publicly available. They're open source. And so these datasets comprise both cancer tissues as well as matched normal tissues. So by using these MMD datasets as well as matching TCGA datasets, we found very strong correlations between metrisome and immune response. So for more details, please refer to these two studies. So here again, what we have found is that we indeed found a very high inter-cancer heterogeneity in terms of this metrisome expression. So although we have observed a very high level of inter-tumor heterogeneity and inter-cancer heterogeneity, it is the intra-tumor heterogeneity, which means heterogeneity within the same tumor, that drives the disease progression and is associated with drug resistance. So here again, we found several publicly available datasets that have multi-region RNA-seq data. So here they have multiple sectors from the same tumor, and these individual sectors are then subjected to gene expression profiling, and the resulting data are then used to compute this TMI. And so here, as we can see from the score distribution, this metrisome scoring, the gray color represents the matched normal lung, the yellow represents the tumor. When we use the cutoff point of 13.3 here, we can clearly differentiate the tumor from normal with 100% accuracy just using these 29 metrisome gene expression. And again, when we just use the tumor samples, and when we use the cutoff here and stratify the patients into two groups, into low and high risk group, these two risk groups have significantly different survival outcomes again. So here the hazard ratio is 5.63. So although we have validated the diagnostic and prognostic accuracy of TMI, we also observed a very high level of intra-tumor heterogeneity. So here the graph shows the x-axis is a patient ID, the y-axis represents the score, TMI, the risk score. And if you look at each dot, which represents the TMI of individual sector, there is a very wide variability in terms of TMI. So here the red boxes represents a patient having discordant tumor samples, meaning that this patient has both high risk as well as low risk tumor sectors. And this was again validated by another independent data set. So all this information or our data clearly indicate that depending on where we obtain the tumor sectors, the patient may be misclassified to, for example, either high risk or low risk. So there is clearly a better genomic strategy needed to improve this patient stratification and further to refine the prognostication. So we then hypothesized that maybe liquid biopsy, which is more readily accessible than tumor tissue biopsy, can be used as a tool to further refine the prognostic gene panel. So here we hypothesize that this abnormal matrixome expression observed in primary tumors may be somehow reflected at later steps of metastasis, which is during the circulation. So here we assessed circulating tumor cells, CTCs, at the single cell level. Of course, looking at CTCs, particularly at the single cell level, because we initially we wanted to analyze these cells and study the cell-to-cell heterogeneity. This whole process may be very challenging. First, because CTCs are inherently extremely rare and fragile. And second is because it's highly challenging to isolate and analyze these CTCs technically. There are, in fact, many ways to isolate these rare cells. So in blood of a cancer patient, there are leukocytes, white blood cells, RBCs, and cancer cells derived from epithelial tissues. And there's only about several tens of CTCs compared to millions to billions of blood cells. So because these cancer cells are derived from epithelial tissue, the most common method relies on the use of immunomagnetic antibodies targeting the epithelial cancer cell surface. However, as disease progresses, these epithelial cells become more mesenchymal, and so they lose these epithelial characteristics. So if we use the conventional method to isolate these rare cells, we may also lose a subset of CTC subpopulations that may have undergone this EMT process. So as a second generation CTC research, technologies utilizing the biophysical properties have been increasingly developed. So as you can see from here, for example, CTCs are generally larger and stiffer than normal blood cells, as we can see from this size distribution plot. So we can use these biophysical properties of CTCs, such as cell size, cell stiffness density, to effectively isolate these CTCs from the normal blood cells. So as physical methods, these antibody-free biophysical characteristic-dependent methods, technologies have involved membrane filtration, chip filtration, fecal gradient, centrifugal forces, and dielectrophoresis. And one of our pioneering efforts in applying the centrifugal forces to liquid biopsy was the use of spiral microfluidics. So our lab has many years of expertise in CTCs and liquid biopsy. So we have been developing a suite of microfluidic devices to effectively these large CTCs compared to, as opposed to smaller normal blood cells. So this can be done in a phenomenon called thin flow fractionation. So in a spiral microfluidics or spiral microfluidic channels, these cells experience two types of forces, inertial force or hydrodynamic lift force, as well as Dirac force, which is induced by secondary flow. So both of these forces are dependent on the cell size. So by modulating these dimensions of the microfluidic channels, we can effectively isolate these microparticles or cells of different sizes as opposed to smaller microparticles. And this is another microfluidic chip that has been recently developed. So it has been developed specifically to isolate cells as single cells. So this device has 10 individual microchannels where each microchannel can capture one single CTC. So here, the figures show the captured lung cancer cells that are positive for nucleus, as well as cytokeratin, CK, which confirms the epithelial cell origin. So using the spiral microfluidics, as well as this microfluidic chip, we have developed this integrated workflow where we can analyze or where we can isolate and analyze patient-derived CTCs at the single cell level for downstream genomic analysis. So I'll briefly go through the steps here. So a 7.5 ml of blood is collected from a patient. So here again, we have studied the lung cancer patient. And this sample is now processed with the spiral microfluidics. And our spiral microfluidics is currently developed as a fully automated system and we name it as a ClearCell FX. So using the ClearCell FX, we can get the output with the CTCs, but because of their extreme rarity, there may be a small number of leukocytes that can contaminate the output. So we can then again use this microfluidic chip that can capture single cells. So here we confirm the CTC by immunofluorescence staining by using nucleus and CD45, which stands for the white blood cell. So if it's CD45 negative and Hox positive, then we think that the cell captured in the microfluidic channel is CTC. So this single cell is then transferred to a PCR tube for downstream analysis. And together with the expression profiles as well as the multi-region tumor expression profiles, which can tell us the information on intratumor heterogeneity, as well as the clinical information such as survival outcomes, we can then refine the initial prognostic gene panel. So here, we next hypothesized that the tissue-derived biomarkers, which was metrisome gene expression levels, we hypothesized that we assessed if these tissue-derived biomarkers would also be differentially expressed in CTCs. So in total, we analyzed 61 single CTCs from 20 Asian NSCLC patients. And as we can see from this heat map, we found a subset of genes that are differentially expressed in patients with distant metastasis as compared to those without distant metastasis. And very interestingly, we also found that high risk tumor compared to the low risk, they had low heterogeneity in terms of metrisome gene expression in both tumor tissue biopsies as well as liquid biopsies. So here we conclude that this metrisome heterogeneity observed in tissue biopsy may be reflected in liquid biopsies as well, which are CTCs. So altogether, we found a subset of gene signatures that are consistently expressed in tumor tissues and are associated with disease progression, poor prognosis, low intratumor heterogeneity, as well as differentially expressed in CTCs. So here we refined the initial prognostic 29 gene panel to only include these MMP genes to further rebuild the scoring metrics based on the expression levels of these two genes. So these are the results. So when we use the refined classifier, we see that the number of patients having discordant samples has been significantly reduced as compared to the initial classifier. We still observed a small number of patients having discordant samples, but generally the level of heterogeneity or the level of variability of this risk score has been reduced. And more importantly, when we performed the survival analysis, we found that this very small gene panel can achieve higher accuracy in predicting the recurrence-free survival. So here the hazard ratio is 9.6 as compared to 5.63 as in the case for the initial classifier. And so we finally went on to assess if we can identify the universal cutoff point. So this would be clinically useful when we have a predefined cutoff as well as gene scoring metrics and the known platform to profile the gene expression. We can then use all these settings to apply to a prospective patient cohort. So here again, we have used publicly available data sets and found a universal cutoff. So here we found that it's 1.441. So when we apply this cutoff index and apply to independent data sets, we can then see that these two groups stratified by this cutoff have a significant difference in terms of recurrence-free survival. The prognostic performance was also observed or validated in multiple independent data sets as well. So here we have validated their performance for overall survival as well as recurrence-free survival. So I hope I have demonstrated how we can address this intratumor heterogeneity as well as CTC cellular heterogeneity to achieve improved prognostication. For more information on intertumor heterogeneity and intercancer heterogeneity in terms of this metrisome expression, you can refer to these studies. So TMI signatures, in conclusion, are clinically useful protumoral and ECM-related genes. And we have also developed a microfluidic integrated workflow to isolate and analyze patient-derived CTCs at the single-cell level. And finally, we have shown the clinical value of these single CTC-derived gene signatures. So of course, our studies can raise more questions than answers. So it still needs to be investigated whether these microfluidic approaches really capture all CTC populations and whether there exist benign tumor cells that and not just aggressive and metastatic CTCs that can survive in the bloodstream, although we think that this is not the case. And there may be other factors that may affect the tumor heterogeneity as well as its impact on prognostication, such as tumor secreted factors, tumor shed EVs, and exosomes. So with that, I'd like to thank my advisors, Prof. Lim and Dr. Darren, and other researchers at National University of Singapore. I'll happy to have any questions.